All right, everybody. Today we're going to be looking at meiosis. So just a quick review. DNA is deoxyribonucleic acid. It's a material that contains information that determines inherited characteristics found in the nucleus of eukaryotes. Proteins are the chemicals that work for your cells. Proteins are what DNA codes for and what are made at ribosomes. Genetics is the study of heredity, and heredity is the biological process whereby genetic factors are transmitted from one generation to the next. So on the right is a picture of human chromosomes. We have 46 per cell. You get half from your mom, half from your dad. And we're a mix of DNA from both parents. So you get an equal mix of chromosomes from both parents. Up on this slide, we've got a picture of a chromosome. Chromosome is a thread-like structure within the nucleus. It's what contains the genetic information that gets passed on from one generation of cells to the next. Homologous is referring to chromosomes that are joined, so pairs of chromosomes joined together by the centromere. On the right-hand side, you've got a chromosome. So if you look at the whole X structure, that's a chromosome. It's two threads. Each one is called a chromatid. It's held together by a centromere. And then if you were to zoom in on that chromosome, you'd see a gene, which is just a segment of DNA that codes for a trait. And if you were to take that chromosome and zoom in really close, it's just DNA that's been wound. So it's a long strip of DNA that's been wound around proteins. So what I would do at this point is pause the slide, take some time to draw out the structure of a chromosome, use that blank side of your note page to draw out the chromosome, and then just hit play and come back in after you've drawn out the chromosome. All right, welcome back. So we're going to be looking at meiosis, which is division of cells that is just for producing reproductive cells in sexually reproducing organisms. It makes 4 cells. Each one of those cells has half the genetic material. And if you think back and remember back to mitosis, it does prophase, metaphase, anaphase, and telophase 2 times. On the right-hand side is an overview of meiosis. You can take a glance at it now if you want to pause the slide again just to look at it. And then you'll get another look at that full set for meiosis later on in the presentation. I want to distinguish between 2 types of cells. Diploid cells is the term used to refer to a cell that contains both sets of homologous chromosomes. And so it's twice the number of chromosomes. So if it was a human being, you have 23 chromosomes, and you have twice that number, so you have 46. A haploid cell would be a cell of an organism having a single set of chromosomes. So gametes in human beings, it's a haploid cell, so it just has 23 chromosomes. Your gametes are your egg or sperm, depending if you're a man or woman. And the end result of mitosis is a diploid cell. End result of meiosis is a haploid cell. And then on your right-hand side, you could see for haploid, you've got 3 chromosomes there. And for your diploid cell below, you've got your diploid cell with your sets of chromosomes. Okay, so the whole process of meiosis to produce those haploid gametes, if you look on the right-hand side, the end result for male would be sperm. End result for female is 1 egg, and then there's 3 other cells called polar bodies. They're much smaller, and they end up disappearing. So just the 1 single egg is produced after a run of meiosis in a female. So you want to take a diploid cell. The purpose is to take that diploid cell converted into a haploid cell. It causes a change in the genetic information. There's also changes in the genetic information to increase diversity. And we'll see that in the coming slides about how chromosomes can exchange information. But again, we take one diploid cell and it becomes 4 haploid cells. So the exchanging of information comes in prophase 1 and metaphase 1. And you can see examples on the right-hand side. So on the right-hand side, you're going to be seeing crossing over. So what that is the actual chromosomes are going to overlay over the top of one another, and then there's going to be a break, and they're going to refuse together. So chromosomes were copied just like in mitosis during prophase 1. They condense and join up as a tetrad. So that's the picture in the lower right, where you've got your pair of chromosomes joined up and crossed over each other. So that crossing over is the exchange of genetic material between the homologous chromosomes. And what that does is it allows for DNA that wasn't existing on a chromosome to switch over now. So the chromosomes aren't exactly like the parent chromosome. Then the chromatids line up in the middle of the cell as the tetrad. The 4 chromatids in a pair of homologous chromosomes that come together during meiosis. And then there's a 50% chance whether the chromosome is going to go towards one pole or the other pole inside the cell. So then we've got meiosis continuing. Anaphase, just like in mitosis, the tetrad is going to split. The chromatids move to each side of the cell. In telophase, the cell's ramping up and preparing for meiosis 2, and then the cell's going to divide. So at the end, we've got two cells forming. But because we're going all the way to 4 cells with half the number of the genetic information, we go through prophase 2, metaphase 2, anaphase 2, and telophase 2, and then again in cytokinesis. If you look at the chromosomes here, you can see that crossing over has occurred. So this piece of the chromosome from here has been exchanged. So we've got this new novel chromosome. They line up, they move away to the other side, and then we produce these 4 cells that are the gametes. If this is a male cell, these 4 cells would turn into sperm. In a female, you'd have 1 cell that becomes an egg, and the other 3 polar bodies are going to just dissolve and get taken back up in the body and aren't going to be used in reproduction at all. And that's it. So that wraps us up, taking us all the way through meiosis 1 and meiosis 2. There's a couple things you should remember. You already know already. The steps of meiosis you already know from mitosis. So you're seeing those again. The only thing that's different is that they're happening twice. And the other 2 novel things is the crossing over that occurs and the tetrad that gets formed between the chromosomes. All right, nice job.